Our easi-tab reference materials are lyophilised microorganisms produced under LGC's scope of ISO 17034 accreditation, available either as a tablet inside a 2 ml microtube or as a 2 ml glass vial. To resuscitate either format, simply take 1 ml of sterile microbiological diluent, add it to the vial or tube, and tap gently to mix. Leave for 15 minutes, and it's ready to use in your routine testing. Depending on your application, you can then obtain the appropriate microorganism count by adding whatever quantity of diluent you wish. Once you have the volume and concentration that you want, you can inoculate the suspension onto agar plates or use membrane filtration to test the sample. Another convenient way of resuscitating an easi-tab tablet is to add it directly to any larger volume of liquid that suits you. For example, you can take 1 litre of diluent, add the tablet directly, then agitate and leave for 15 minutes until it's fully dissolved. This can now be used with agar plates or membrane filtration in the same way. Each shipment of easi-tab reference materials contains a detailed instruction sheet that outlines the 2 resuscitation methods. It's also available online at lgcstandards.com/easi-tab. In addition, we provide a detailed statement of measurement that describes the strain present in the easi-tab, its reference value and expected measurement range, and date of production. easi-tab reference materials that fall under the scope of our ISO 17034 accreditation also include the UCAS accreditation mark on their statement. To ensure their stability, easi-tab products should be stored in the freezer as soon as you receive them, and most can be used for up to 2 years after their production date. To find out more about the easi-tab range, or to place an order, go to lgcstandards.com/easi-tab. And to learn about LGC Axio's range of microbiology proficiency testing schemes, search lgcstandards.com/axio. LGC. Science for a safer world.